Hello students, welcome to Simplify Pharma. Please do subscribe to my channel to remain updated with the latest video lectures. We already covered gene therapy for cancer part 1 and now in continuation in this lecture we will be studying gene transfer strategies for treatment of cancer. As we saw in the last lecture, cancer is nothing but uncontrolled cell growth. There are several gene transfer strategies for treatment of cancer, such as modification of tumor cells, sensitization of normal tissues or tumor cells, modulation of tumor invasiveness, enhancement of the anti-tumor immune response, that is also called as immunotherapy, Oncolytic Virotherapy, gene transfer therapy. So overall there are 6 gene transfer strategies for the treatment of cancer, and we are going to study each category in detail one by one. Starting with modification of tumor cells. Repairing genetic defects is believed to be responsible for tumoral proliferation. Now what are they trying to say? Many times we use gene therapy to repair the genetic defects by replacing the altered gene with the properly functioning gene. But this repairing of the genetic defect, this is said in many cases, in many clinical trials, this was found to be responsible for cancer. Now for example, by restoring the genes controlling cellular division or that induce programmed cell death apoptosis, it results in proliferation of tumor. In theory, gene therapy could be used to replace an inactive gene with an active one or to neutralize an abnormal function gained by a mutated gene. Inactivation of regulatory genes frequently induces tumor cell growth. For example, mutations of the p53 gene have been identified in numerous tumor models. The exact role of the p53 gene has not been totally identified, but it has been found that it suppresses the genes involved in cellular proliferation, and it can also induce apoptosis. So restoration of the activity of p53 gene in the p53 mutated or deficient tumors, it may stop the uncontrolled cell growth, or it can also induce apoptosis. And several strategies were used for restoration of the activity of p53 genes, such as using adenovirus, retrovirus, or non-viral p53 gene transfer. Clinical trials have demonstrated the potency of p53 gene transfer in several carcinomas like head, neck, lung. Now in this diagram we can see when the gene was induced with one of the vector, and then this gene was incorporated inside the tumor cell, which led to apoptosis of the tumor cell and thus lysis and engulfing by the macrophages. The second approach that we are going to study in detail is sensitization of normal tissues or tumor cells. Introducing into the tumor cells genes encoding for an enzyme that can transform a non-toxic prodrug into an active drug, or by introducing genes into normal tissues that can protect them against the effect of anti-tumor toxic drugs. Now they are saying there are 2 ways by which we can work in this approach. The first thing is by sensitizing the normal tissues or by sensitizing the tumor cells. Now how can we sensitize the tumor cells? If we introduce genes inside the tumor cells that encode for an enzyme and that can transform a non-toxic prodrug into an active toxic drug, and such genes, they express themselves only in tumor cells. By this we can get targeted drug delivery, and thus we can save the normal cells from the adverse effect. There's one more approach in which we sensitize the normal tissues, like we introduce the genes into the normal tissues that can protect them against the effect of against the toxic effects of the anti-tumor drugs. So these are the two approaches: prodrug-metabolizing enzyme and cytotoxic drug resistance gene transfer. Now for that we need to understand the difference between prodrug and active drug. Now prodrug is inactive before metabolism. As soon as it reaches the liver, it becomes active. That is, it becomes active after metabolism. Whereas on the other hand, active drug, the moment it reaches inside the body, it gets activated and it starts showing its effect. So prodrug doesn't show its effect unless and until it becomes active, whereas the active drug, the moment it reaches inside the human body, it starts showing its effect. So discussing the first approach in detail, prodrug-metabolizing enzyme. In this concept, the gene encoding the prodrug-metabolizing enzyme is expressed only on the surface of cancer cell, and it metabolizes a small molecule to an active moiety, which then kills the tumor cell directly. The molecule may also diffuse either through intercellular gap junction or in the extracellular space and destroy adjacent tumor cells. So in this way, what are we doing? Transduction of even a small proportion of tumor cells can produce a large bystander effect on adjacent tumor tissues. Now, what is the major limitation of this approach? It requires local inoculation of a tumor with the vector encoding the gene. So using this approach, we can only target the tumor locally. So all the tumors that are restricted to a particular area, only those tumors can be targeted using this approach. The metastatic tumors, they cannot be targeted using this approach. Now let's take few examples. The brain tumors, they were an attractive initial target for this type of gene therapy, because these tumors, they very rarely metastasize. The goal of the therapy is local eradication of the tumor. So more recently, a study has been performed on patients with retinoblastoma, which is also a highly localized tumor that is conventionally treated by enucleation or chemoradiotherapy. Now, as we all are aware, enucleation is disabling and deforming, and if the tumor is bilateral, it leads to blindness. Enucleation, if the students are not clear with this concept, it is a surgical procedure that involves removal of the entire globe and its intraocular contents with preservation of all other periorbital and orbital structures. In simple language, surgical removal of eye. And this type of ocular surgery is indicated for a number of ocular tumors in eyes that have suffered severe trauma and in eyes that are otherwise blind and painful. Now coming to the next approach, cytotoxic drug resistance gene transfer. Now using this approach, if anyhow we sensitize the normal cells and make them resistant to the toxic effects of anti-tumor drugs, in that way we can give high doses of anti-tumor drugs to the patient, and that will target only the tumor cells, sparing the normal cells. So there won't be any side effect of the anti-tumor drug, any cytotoxic side effect. And in this way, we can target and kill the tumor cell. Now, by increasing therapeutic index in this way, it is hoped that more drug can be administered and higher percentage of patients can be cured. But obviously, it will also have certain problems. The lack of targeting of current vectors means that they may transduce malignant cells and normal cells too, and therefore increase the resistance of both to the cytotoxic drug. So it can happen that along with the normal cells, we are also sensitizing the tumor cells and making them resistant to the anti-tumor drug. This is the one disadvantage. The other one is, although it may be possible to protect a significant proportion of marrow stem cells, secondary toxicities to other organ systems such as skin, lung, and gut will rapidly become evident as doses are escalated, because these tissues are much less readily protected than hematopoietic stem cells. Now, if we increase the dose of anti-tumor cells, though the normal cells are not affected because we are sensitizing them and they become resistant to the anti-tumor drug, but there can be several other organs, that can get affected due to the high level of anti-tumor drugs, the toxicity that it produces. Coming to the third approach, modulation of tumor invasiveness, in that antiangiogenesis gene therapy. Now, in order to understand antiangiogenesis, we need to understand the term angiogenesis. Angiogenesis is nothing but the formation of new blood vessels in simple language. This process involves migration, growth, and differentiation of endothelial cells which line the inside wall of blood vessels. And this process is controlled by chemical signals in the body, such as vascular endothelial growth factor (VEGF) that bind to receptors on the surface of normal endothelial cells. When this VEGF and other endothelial growth factors bind to their receptors on endothelial cells, signals within these cells are initiated that promote the growth and survival of new blood vessels. So we can see over here, VEGF and VEGFR. These are the activators that activate the endothelial cells and that initiate growth of new blood vessels. And these are the inhibitors, endostatin. If there are growth of new blood vessels, then the tumor cells will replicate even faster. This is how it happens. Now, what is the role of this angiogenesis in cancer? This is very important concept that we need to understand. Only then we will be able to understand the anti-cancer approach. Angiogenesis plays a critical role in growth of cancer because solid tumors, they need blood supply if they are to grow beyond a few millimeters in size. Tumors can actually cause this blood supply to form by giving off chemical signals that stimulate angiogenesis. And one very interesting fact, tumors can also stimulate nearby normal cells to produce angiogenesis signaling molecules. The resulting new blood vessels, they feed the growing tumors with oxygen and nutrients, allowing the tumor to enlarge and the cancer cells to invade nearby tissue, to move throughout the body and to form new colonies of cancer cells. This is called as metastasis. Because tumors cannot grow beyond a certain size or spread without a blood supply, so scientists are developing drugs called as angiogenesis inhibitors which block the tumor angiogenesis. The goal of these drugs, also called as antiangiogenic agents, is to prevent or slow the growth of cancer by starving it of its needed blood supply. So in short, the cancer cells, they require more oxygen, they require more nutrients to feed themselves. Only then they grow, because their growth rate is more than the normal growth rate of the cell. That's why their needs also enhance. Now, in order to feed themselves with this enhanced need of oxygen and nutrient, they require extra blood. And how do they get it? By growing new blood vessels. And thus this process is called angiogenesis. So anyhow, if we can prevent or slow down the growth of these new blood supply, the cancer cells will starve to death. So how do these angiogenesis inhibitors work? They are unique cancer fighting agents because they block the growth of blood vessels that support tumor growth rather than blocking the growth of tumor cells themselves. If we cut the supply of blood, it will starve the cancer cells to death. Angiogenesis inhibitors interfere in several ways with various steps in blood vessel growth. Some are monoclonal antibodies that specifically recognize and bind to VEGF. When VEGF is attached to these drugs, it is unable to activate the VEGF receptor. If it doesn't get activated, the signal won't be passed for the formation of new blood vessels. Some angiogenesis inhibitors are also immunomodulatory drugs, agents that stimulate or suppress the immune system, that also have antiangiogenic properties. What angiogenesis inhibitors are being used to treat cancer in humans? The US FDA has approved a number of angiogenesis inhibitors to treat cancer. Most of these are targeted therapies that were developed specifically to target VEGF and its receptors, or other specific molecules involved in angiogenesis. This is the list of approved angiogenesis inhibitors. Coming to the next approach, this approach that we were studying in detail. Now we can study this approach because we understood the meaning, importance of angiogenesis. Modulation of tumor invasiveness, antiangiogenesis gene therapy, delivering the genes that can inhibit the growth of new blood vessels to impede nutrient supply to the tumor cells. That is the inhibition of new angiogenesis. Now, antiangiogenic gene therapy. There are several recent developments in that, wherein we can see various nanoparticles are used. Now, in this diagram, what we can see over here, that there are several approaches that are used to block the angiogenic growth factors, that is VEGF, FGF, PDGF. These all are angiogenic growth factors. So if anyhow we block these angiogenic growth factors, we can block the mechanism of angiogenesis. So we can see in this example there is an endothelial cell, and on the cell surface there are several receptors. Now this figure depicts different modes of gene therapy directed to tumor endothelial cells and its microenvironment. The expression of endothelial cells specific cell surface molecules like vascular endothelial growth factor receptor VEGFR, E-selectin or angiogenic growth factors VEGF, FGF, PDGF produced by tumor cells can be inhibited by specific antibodies or antisense RNA or using gene-specific siRNA. The targeted gene therapy can be achieved by using either viral vectors or nanoparticles carrying ligand to endothelial cell surface specific receptors. To target the angiogenic genes to tumor vasculature, the inhibitors can also selectively express in EC using endothelial cells specific promoters like endothelial 1, now this anti-angiogenic therapy, there's several possible mechanisms. For example, the tumor does not respond to VEGF blockade. then in such case we can use anti-angiogenic agents that target alternative pathways. If it is ineffective, or there is no in combination cytotoxic therapy, then the possible solution is combine the anti-angiogenic agent with effective cytotoxic therapy. If inefficient vasculature acquire resistance to the anti-angiogenic or cytotoxic agent, then what can be done is modify the dose or schedule of anti-angiogenic agent, or use alternative anti-angiogenic or cytotoxic agents. So these are the several possible mechanisms by which the tumor can escape the anti-angiogenic therapy, and that can also lead to recurrent tumor growth, but these are also the possible solutions for that. Coming to the next approach, and very important approach, is immunotherapy. In this lecture we will cover immunotherapy in short, and very soon I will upload a next lecture in which we will be covering immunotherapy in detail. Inducing the recognition of tumor cells by the host immune system, or by enhancing the cytotoxic function of immune effectors. So what actually we are doing in immunotherapy, we are activating the host immune system so that it recognizes the tumor cells and kill it. So basically we use genetically modified cells and viral particles to stimulate the immune system to destroy cancer cells. One prominently used is lung cancer. There are the three different pathways that can be used. The first pathway is in which immunotherapy is using altered cancer cells. The cancer cells, they are altered with the genes, and they are injected inside the human body, and thus it activates the immune system, and that lead to tumor cell destruction. There is second pathway that represents the immunotherapy with genes in vivo, wherein the genes, they are injected inside the cancerous tissue, that lead to activation of the immune system, and thus the immune cells, they can find the cancer cells easily and then thus destroy them. There is one more approach in which immunotherapy is used by altering the immune cells. The immune cells, they are altered with the gene then that can specifically find the tumor cells by activating the immune system, and thus the tumor cells are killed. Now normal white blood cells are removed from the cancer patient. These white blood cells are infected with viral vector designed to deliver genes into cells, and these genes enable the white blood cells to make T cell receptors that stick to cancer cells, and after the white blood cells are reintroduced into the patient, the T cells bind with the cancer cells and destroy them. Coming to the next approach, oncolytic virotherapy. In this we use the viral particles, that is the viral vectors that replicate within the cancer cell, and it causes cell death. Example are metastatic cancers. So over here we have genetically modified viral vector that is injected inside the cancerous tissue that lead to cancer cell lysis and destruction. Last approach is gene transfer therapy. It introduces new genes into a cancerous cell or the surrounding tissue to cause cell death or slow the growth of cancer. Examples are in solid tumors. So the genetically modified genes, they are introduced into the cancerous cell, wherein they replicate, and that lead to cancer cell lysis as well as destruction. These are the several examples of the cancer type and the drugs that are there in the market and the various approach that they have used. As you can see, for prostate cancer, there is already a drug in the market that is using trichome vector vaccine, and it is using the concept of immunotherapy. Then there is something for soft tissue sarcoma cancer, in which adenovirus vector has been used and the approach is gene transfer. Then there is something for the pancreatic cancer, wherein the retroviral vector approach is used, and this type of therapy is again just gene transfer. This approach is based upon the gene transfer therapy. Now there are several challenges in gene therapy, like identify more efficient ways to deliver the genes to the patient genetic material. We do have several ways like gene gun, we directly inject them, but these are not that efficient. Second is deliver the genes to precise location in patient's DNA. Again, this is very difficult to do. Develop vectors that can specifically focus on the targeted cells, so that we can reduce the side effects of the cytotoxic drugs on the normal cell. Ensure that the vectors will successfully insert the desired gene into each of the target cell. Now this is something that happens in vivo, so it is very difficult to ensure it while sitting outside. So this is the reason we require vectors in which the success rate is pretty high. Then ensure that transplanted genes are precisely controlled by the body's normal physiologic signal. It should not happen the transplanted genes, they are rejected by the body's physiologic system. Thank you. Stay tuned to my channel for further video lectures and do subscribe. In the next lecture, we are going to cover immunotherapy in detail. Thank you.